Is it working? Let us see. Hello, 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 hello. Is it working? Yes, it is. Okay, cool. First things first, let's do our Duolingo. Greek. Got my keyboard ready this time. Right, forms of S. I think I did a bit more since you're gone, but doesn't matter anyway. So, os. That one. And it's this sigma, I think. Oh, snow. Okay, so snow would be this one. Sno. Match the pairs. Sno would be sno. Ria, ria. Os, os. Ooh, that's a good one. C? V. I think it's V. Aye, aye, aye. Guess it's that. Oh dear. Okay, meh, meh. V, V. That's interesting. Oh, because that's the English pronunciation, I see. Excuse me, sorry. Oz. Okay. Oh, I think I've lost the thing. Aftos kane rizi. Aftos is he, I think. He, yeah. Kane, aftos kane is makes rice. Rizi. Sno. This one. The snack with uzu. To snack me uzu. Uzo. Okay, I don't know what uzo is. What's uzo? Oh, nice. Is it alcohol? Liquor, yeah, nice. Video, vid, video, ke, snack. Video and snack. The snack. To snack me uzo. The snack. The snack with uzo. The snack with uzo. He makes rice. Okay. Yeah. So this should be aftos. Like that? No, it's not that. It's that one. Aftos? I think that might be a C actually at the end. Yeah, or that thing. Okay, let's do that. Aftos. He? Me? Rizi. Rizi. Something like that. So, autos. Autos. Cane. Rizi. Okay. Oh, I'm not going to remember it. Autos cane rizi. Autos cane video. That's not right. It's video. Thing like that. Vinteo. Like that maybe. Oh, nice. Cool. I got that right. Makes cane autos. Okay, awesome. So autos. Cane. Yeah. Rizi. I think I've done something wrong here. Autos cane rizi. Oh, I should trust myself more. Awesome. Another one bites the dust. I'm trying to hold off a sneeze, so my apologies. 1 second. Nice. Okay, cool. I've probably got like tissue on my moustache now. That's done. Okay, right. What are we doing? So we are looking at some of the previous stuff because I think we were just starting to explore. Uh, we were just starting to explore AI and the formulas used in AI, and also how I could use it to help, like use AI to help me discover some interesting features of the nervous system, or at least one idea of it. And let's see, zoom in here. Uh, we figured out there was a protein data bank, which means I can take existing data or DNA sequence and try and make it through AI essentially. And then what else we got here? This paper. We got here on the paper. So what was this about? I don't remember. Let's have a look. Maybe it's already open actually. Let me try and close some of this stuff. Oh yeah, here it is. Okay. So I have to remind myself what this was for. And we are at page 708. Improved protein structure prediction using potentials from deep learning. Protein structure prediction. Oh, that's right. I think we were looking at AlphaFold, and like how they did it. So yeah, that's all I've got here. Our training, validation, and test data splits. Cath domain codes are available from GitHub. Oh, DeepMind. Yeah, that's right. So we're looking. So DeepMind, their AlphaFold, I think not the latest version, but the last version was available on GitHub. So we were starting to have a look through that. And they mentioned that they used Protein Data Bank, and they have all the code there, and they had this paper to go along with it. So let me just look at the abstract again because I can't quite remember what that was saying. And I have way too much open here. Let's go back over here. Yeah, we'll have to go through all that stuff anyway, so yeah, okay. Back propagation, cool. Yep. Fast and simple distributed computing. That was like that was like a mention in the PyTorch documentation, I think. Desmos, Rayleigh, differentiation. Some look at Menelaus-Kellis stuff. Here it is, right. DeepMind. AlphaFold. Awesome, awesome. Oh, yikes, okay. Oh, oh yeah, I see. So this was this competition that they were doing. And it was comparing to all the other participants, I think. That was cool. CASP 13. So they're on CASP 14, I think. That's right. Yeah, CASP 14 is the new one. Okay. Stochastic having a random probability distribution or pattern that may be analyzed statistically but may not be predicted precisely. Cool. The von Mises distribution, probability theory and directional statistics. The von Mises distribution, also known as the circular normal distribution or Tikhonov distribution, is a continuous probability distribution on the circle. Cool. Uh, Van der Waals, force include attraction. Okay. Right, okay. So let me see. Have I got any other notes here? Is that all I left? Hmm. Yeah. Okay, that's a shame. Didn't really leave much of a scent, did I? Should have really watched the last video I made. Okay, I guess what we should probably do then is start again with this AlphaFold thing, because okay, so once again I keep saying it, but the idea is to um so number one, my apologies, I'm a little bit tired, but I thought I really wanted to give this a go again. So the idea is to take okay, yeah. Let me just actually explain this properly. So the idea of all of this stuff I'm doing is to take, let's say, a standard or let's say a happy human, like a selection and the variety of happy humans. Happy. So that might denote, in terms of the goals of this project, happy might denote that there is the absence of major disabilities, things like diabetes or cancers or um, uh, badly functioning hearts and stuff like that. Things that really disable you. If you are to compare yourself to a a biological mechanism that has more potential in terms of like mechanical output or longevity of life and all that stuff. So um, I understand it's a bit weird, but I'm trying to very very rough. S equals happy. Okay, whatever that means. And let's say you have a um, like a set. Let's say let's call them D for disease for now, because disease is a good one to go for. Let's call them D. And you might have disease 1, and you might have disease 2, and probably disease through to N. um, so these are diseases. And what we want to do is learn more about uh, the differences between S and D to DN, as like using S as a training model and D as the comparative data. I think that would be interesting to see what AI can produce for us, in terms of um, finding out specific differences, or maybe you could say this is a collection of people who have Alzheimer's, or this is a collection of people who have diabetes. Take the state of a human, compare it, and find the differences. Cool. So I think that's relatively good reason for AI to be here. And if if we go back a little step or a step further after that, the idea is not particularly to solve the problems of what cures these things, but it's more to do with understanding the body as a whole. So if we were to know or identify portions that represent disease, then when we grow a greater understanding of them. Now this is work that's being done. Correct me if I'm wrong, Manolis Kellis, I apologize, but the University of MIT, Massachusetts Institutes of Technology, I think that they're doing work on identifying um, the hierarchical, identifying the hierarchical dependence of um, uh, the gene of the DNA sequence. So something like that. So essentially, I think they're already trying to identify what part of a genetic sequence belongs to diseases. So this is fine. We're not really trying to do this, but we are trying to understand at least one step towards a goal, and that goal is if we have our Earth and a comet comes in and we don't have any more options, then I want us to be able to send off little, genetically modified beings, and they are genetically modified with like biological buffers to prevent damage from, let's say, the radiation of space travel, or the if your rocket was to explode, you would be able to I don't know, survive in extreme coldness, or you I don't know, your rocket blows up, so then you need to make sure that you're not going to die from heat. Oddly, I'll put that in as green because I don't seem to have any other colours. So all of these things, we might want to try and put a buffer around the biological mechanisms in our body, so that we can indeed get to places like Kepler and other planets with good good means of life, etc. So that as a humanity, at least a somewhat genetically modified humanity, we can continue to exist. So I suppose that would be the largest goal. But on a route to that, we really want, or I really want to be able to solve some of these diseases. And I just find that perhaps if we are getting this as our bigger goal, then we are probably solving some of at least these problems. So that's the hope. And so where does AI come in? Well, AI comes in by allowing us to vigorously test against data we already have. Let's say protein folds like this, and we have some AI that we give it some randomized data, and we try to reproduce this protein and its way of folding with the process of AI. And so once we get this about right, it's important to understand that this is a protein, and a protein and the way it's folded denotes its function in the biological being. So let's say this one might, I know, break down, or it might, this and a collection of other things might, I don't know, make your eye blue, or another one might, I don't know, make your toe slightly big. I don't know, things like that, right? So the fact that these things, these proteins and the way they fold, denote their function in our body, but that more importantly, or more interestingly, a DNA and its nucleotides, I think that's what they're called, these nucleotides, or their sequence of like A, G, whatever it is, etc., etc., they denote how a protein folds, and thus what the function of that protein is. So this is AI, and this is where it's going to come in. And so this whole thing is important to understand it. And just as a side note, because there's a lot to unpack there, I had originally had people in my family who had disabilities that prevented them from walking. So you had, I think it was motor neuron disease, something like that. And then we have or muscular dystrophy. No, motor neuron. and then you had cerebral palsy, so motor neurone, cerebral palsy, and of course these guys in my family, they cannot walk. Um, so I had originally wanted to make some kind of exoskeleton to, use Tesla's kind of guidance software, and there'd be cameras, and then you'd have like Neuralink, and of course that goes in. Then you've got this beautiful human being here who uses Neuralink to guide where it's going to walk. And of course, maybe their body doesn't allow them to actually do the walking, but the exoskeleton certainly could. So that was the original idea, idea one. But this has a problem because let's say if you're thinking about in the future, there is a likelihood, at least a significantly non-zero likelihood, that people aren't going to want to be so thinking so much of their bodies, more so their mind. If you take things like Neuralink, let's imagine it goes into the brain, and you can control things with your mind. Well, maybe you could write vision into the mind, and so the reality around us is less significant to us because we are creating, our beautiful worlds inside our minds, and we can communicate telepathically and all that stuff, back and forth. What's the point in having a body that can walk and work if you can already do it inside your mind? So that's the question. Is there any point in doing this? Well, probably not. And here's another one. If you were to try and make yourself last longer, you want to go interstellar, then you might start to put metal interfaces and like literal pieces of material that are not biological material, and you might put your brain onto a chip, and all those kind of things. And that's great, and that might allow you to go interstellar. But we have some problems. Is that Neuralink might be interfacing with the brain, but I have this. I have a hunch that there will need to be some kind of biological modifications to enable this to really work. If you're putting in electrodes into the brain, a famous phrase was once the brain's seen air, it's never no. If the brain's seen air, it's never the same, or something like that. I feel like, as wonderful as this is, it's pretty invasive, and also these do not last forever, at least currently, and so on and so forth. Many things that I might be able to support, given a modified genetic sequence that will allow better uptake of such things, or and this is the goal, again, back to this AI, this interstellar travel for genetically modified humans, is to actually, so this is my version, is to modify the DNA in such a way that no need for this, no need for this, and you have this resistant DNA modified per place you're going to go. And at the same time, there might be some kind of linking here with Neuralink, because I could maybe give a higher bandwidth of nervous system, or the neurons, as they trigger, it might trigger something else, and then that could be read rather than being read from the brain. So lots to think about. That's the goal, and I thought that was important to say, because I don't know if anyone ever watches this, but I just do stuff, and there's no reason for it if you just tuned in. So here's the goal, and this is why I'm doing it. So with that in mind, let's take a massive step back, and we need to just learn. Let's say over the next 50 years I learn, that's fine. Over the next 100 years I learn, but the goal has to be this stuff, because I don't know how much time we've got before this happens. So Let's go ahead and do at least some of that now. He says we're going to be. So we'll start by having a little brief overview of what AlphaFold was doing. You know what the repository looks like. Lots of Python files I see here, and then we might continue to read this, and maybe if we can digest it together. And you know, it looks pretty long, but maybe if there's enough time, we could skip back onto actually learning how to do AI. Oh, actually, there's one more thing. So Grant Sanderson, who is I think mathematician, I think he might even be from MIT. Not too sure. He has a lovely video on how AI works, and I keep getting it wrong, and I think I tried to explain it last time on this video, but essentially, let's see if I can get it right this time. So I'm going to follow roughly what he does. I'll try my best. Can you see red here? I think you can. It's a bit difficult. It's not scrolling down, unfortunately. I don't really understand how OneNote works. That's the only way I know how to expand the page. Okay, right. So let's imagine that you have some training data, and it comes in to a network, and this network has lots of these neurons like in the brain, and we labeled them. Once again, this is taking it from Grant Sanderson's 3 Blue 1 Brown, just trying to go through it like he did. Let's label these activation, activation, activation. These neurons get activated, and let's say there's a another layer, and these are also neurons, and they are activations. Cool so far. I might get this wrong, but this is the learning process. And I think as this training data comes in, you are let's imagine the training data is a lot of rows in a database table, and they might be like, I know, numbers 100, 200, 400, whatever, and they're data to represent something, and each row represents an A. So here's my activation with its data associated with it. Now, to explain it like Grant Sanderson does, he says that each activation is a pixel, for all intents and purposes, a pixel in a picture, which obviously denotes a number from 0 to 1, where 0 is white and this is black, or whatever. But we might have data like this. Cool. What am I doing? Do we need to do that? Okay, so now we have our activations. Well, these are now numbers, and we can label them as such. Let's call them 1, 2, and 3, so they're not all just A. And this one as well. So let's say this is 1, 2, and 3. And you might be thinking why there's a different kind of set of neurons over here. Well, this is a layer of activations, and so is this. So this is layer 1. So we'll label these layer 1, layer 1, layer 1, and this is, sorry, I didn't mean to do that. See, I would have gone wrong. This is a layer, and this is a layer. Okay, cool. So the data comes through into the activation. This is a number now, and then we are asking the next layer, or we're passing it down through this kind of equation, this function, and the output of that will become the next neuron. So this data 100 might be skewed by some function, and then it becomes something else in here. And so we apply A1 to A1, and we apply A2 to A1, and we apply A3 to A1. I think that's it. And this is called a weighting. So we have, I think what we do is it's A of layer times weight. of layer, and then we do plus the next one, so it would be A1 times weight plus A2 times weight. I don't know what's going wrong with my, I can't click, it's weird. No, I can. No, I can't. Great, it's crashed. Okay, now it's back. Okay. So we have this this activation. We're going to add them together. So this will be A2 in layer times weight in layer, and we keep. I don't know why this is here. And we keep adding them all together, A1, A2, A3 to AN. And then at the end of this, we have to do it now for this one as well. And so you get this equation pretty long. You get lots of these equations, and each one is a kind of function that's doing something, and it's taking an initial value, and it's got this output value, and then this needs to be told, when you did this operation, is the output what I expected? And if it isn't, then you need to tell the computer if it is or isn't the correct output. So, and this is what we call a bias. Once again, this might be wrong. Please go and check this video, and I will definitely keep trying this until I get it right. So the bias is something that you plus at the end. So you have this whole amount of activations, 1, 2, 3, all for this, and then it'll be plus B. And then it'll do the same for the next one, and then plus B. And B can be, -1 to +1, something like that. Let's do that as a B. And then this whole output, I think, is not like in a normalized state. So it might be ranging from various different numbers, very variable. And I think the way they address that problem is they put this through a function. And I think Grant Sanderson mentions it being a squishify function, because it squishes all the values. Let's say you've got this curve here of all your values, and I think it takes like let's say we've got this from 0 to 1 or -1 to 1, and it tries to map all the values into this segment here, so it's like normalized. And I think he uses in his video this sigmoid thing, which apparently is a graph that looks like this. I think it's something like that. And so it uses this graph or equation to squish the data that we've got out of our operation here. Now, this is one way of doing it, but I think there's now a better one that's called Readme. And the difference is quite dramatic. And in fact, if you compare the way it looks, let's say this other thing down here, instead of looking like that, it looks a bit more like that. Readme and Sigmoid. Okay. So that gets applied. And of course, the output of this is the cost equals C. So this is the output of the whole operation. We call this C, or at least Grant does. So this is C, where C stands for cost. And that cost gets put back into the activation layer for the second way round. Now, if we're going to do that, we'll find out. We'll get closer to the right answer over time, and this equation is fine. But Grant then goes on to say that you can do things like back propagation. So if you treat this as like a stepping equation, this would instead become A1L minus A1 A1L, or sorry, plus is A1L times, I can't remember now. It's one of the others. This is really fundamental, and I should really know this, but it's basically L minus 1. So you're going back and adding these up, and that weight goes back and propagates back through this idea of a back propagation. So this stepping is a thing, and of course the weight also gets applied to this. So I think it's referenced in the stepping. So it'd be L and L minus 1 and stuff like that. You see, I've lost my tack here. And of course, it's important also to know that C is the output of this one, but you're passing it into the new one, so you have this kind of stepping indices here as well. I think that's right. So given all of this and my understanding of it, and I think I've got it wrong quite a lot here, maybe there's one more part I forgot to explain, is that this is obviously going to be a really long and annoying equation, so you can compress it into a matrix. So you could say that all from A to A of n, A to A of n, can be compressed into, where it would be A1, A2, A of n. Well, this can be equal to A, which represents the entirety of that output, that series. And the same with the weighting, and the same with the next layer, and the same with the C's and the B's and all that stuff. So we're just compressing them into one variable here. Okay, something like that. So given all this, we've got a lot of detail to actually make sure I've done it correctly. And so I guess maybe we should write down some things we need to really confirm. How do I write something? Type. Here we go. So I don't know how to. Can I do enter? Not quite. Don't know how to. So one would be ensure. Oh, we're still on Greek. Nice. Ensure that we understand the equations correctly, and that I can express them correctly, because the above is likely to be wrong. Ha! 2 is to put the actually do some actual AI work in PyTorch to get a better sense of the above. Revisit and re-explain the above equation with our new understanding. So that's important, right? Because I want to deliver, really clear information, and I still don't understand it enough. But we're going to get there, and I'm going to explain this again and again until it's right. So that's that one, and then we need to also go back to trying to understand what AlphaFold does to some very, very shallow level. Okay, so this might be maybe a picture over a large amount of time, but let's skip ahead here, and we'll go back to 1, 2, and 3 in later videos. Cool. Hi. Okay. So let's read through this, or at least let's try and find out where this all starts. There's a Readme file, and this is the Readme. Great. I'd say this doesn't look like the entire thing. Let's see, there's a Readme here. Okay, so This repository contains implementations and illustrative code to accompany DeepMind publications. I see. So it's not giving us the entire set of code, but more so information that relates to um, information in the papers that will relate back to this and give further clarification. Okay. We release open source environments, datasets and code to enable the broader research community to engage with our work and build upon it, with the ultimate goal of accelerating scientific progress to benefit society. Here. For example, you can build on our implementations of the DeepQ network or differential neural computer, or experiment in the same environments we use for our research, such as DeepMind Lab or StarCraft 2. If you enjoy building tools, environments, software libraries, and other infrastructure of the kind listed below, you can view open positions to work in related areas on our careers page. For a full list of our publications, please see this. Okay. Right. Let's go back to the CASP stuff. And this says, any publication that discloses findings arising from using this source code must cite improved protein structure prediction using potentials from... Okay, so that's cool. The paper abstract can be found here. This is what we're reading. This code can't be used to predict structure of an arbitrary protein sequence. It can be used to predict structure only on the Casp13 dataset. Links below. The feature generation code is tightly coupled to our internal infrastructure as well as external tools. Hence, we are unable to open source it. We give guides as to the features used for those accustomed to computing them below. See also issue 18 for more details. This code works on Linux. We don't support other operating systems. Interesting. Cool. It's very interesting that they use Python. Very interesting. They're using TensorFlow rather than PyTorch. You set up the Python environment. Okay, we could do all this. Alternatively, you can just run the run evaluation shell command. Oh, sh, whatever that is. While the code is licensed under the Apache 2 license, the AlphaFold weights and data are made available for non-commercial use only, under the terms of the Creative Commons Attribution, non-commercial for international, blah blah blah. You can find details at here. You can download the data from here. Let's see what it looks like. That's a license. Dataset to reproduce AlphaFold's Cas13 results. Let's see what it looks like. Downloads. Copy that over to some desktop. Yep. Extract to. Cannot open file as archive. Extract tool. Before you can extract files, you must copy files to this compressed zip folder. To close the wizard, click finish. What? I don't understand. Let me try that again. That's a data set. Save. Download. 7-zip. Extract to. Cannot open as archive. So that's interesting. Let's see. So there's already a problem. Where is this stored? It's a bit.ly, so we won't know. Let's see if there's any hint of it inside the network. Storage, okay, it's a Google API thing, okay. Let's try that. Sure. Huh. Okay, well maybe I can open this. Or maybe I can read on, maybe it'll tell me how to open it. The dataset to reproduce AlphaFold results can be downloaded from here. Cool. The dataset is in a single zip file called zip, which has about 43.5 gigabytes. Oh, that's the problem, I guess. This is 0 bytes, I see. Fine. Hmm. It contains stuff. So actually, I'd like to see this data. Let's see what the weights look like. 7-zip. Same problem. No data. That's a problem. Okay, let's go read their study then, because clearly we're not going to go anywhere with this. I wish this was black background. Oh, it's 9 o'clock. Actually, what I'll do is I'll have some dinner and I'll come back to this. Just realised I haven't had dinner, so let's read this, but I will come back maybe in about an hour or so's time. So see you soon.